Bacterial conjugation, a type of horizontal gene transfer technique in bacteria. The initial evidence for bacterial conjugation was given by Joshua Lederberg and Edward L. Tatum in 1946. They conducted an experiment and through that they proved that transfer of genetic information occurs between bacterial cells when these two cells are in direct contact. He used two auxotrophic strains for the experiment. What is this auxotrophic strains? Bacterial strains, we can classify it into mainly two types, wild type and auxotrophic strain. Wild type strain and auxotrophic strain. Wild type strains means they can synthesize all the nutrients whatever required for their growth. Okay, so we can cultivate them by using a minimal media. Minimal media means the media may not contain all the nutrients. The media what we are preparing, it may not supplemented with all the nutrients. That is what called a minimal media. So if wild type microorganisms are growing in a minimal medium, they will grow normally because they can synthesize all the nutrients. Let us see how Lederberg and Tatum conducted the experiment using the auxotrophic strain. Okay, he used two auxotrophic strain for the experiment. Okay, in the first test tube, you can see there is an auxotrophic strain which cannot synthesize biotin, phenylalanine and cysteine, which is given a negative mark, and it can synthesize threonine, leucine and thiamine, which is given with a positive sign. Okay, in the second auxotrophic strain, the strain which can synthesize biotin, phenylalanine and cysteine, but it cannot synthesize threonine, leucine and thiamine. So in his experiment, what he did initially, he separately cultivated these two auxotrophic strains by using a minimal media, but he found no growth because these auxotrophic strains cannot synthesize some nutrients, so they cannot grow in a minimal media. In the second step, what he did was he just mixed these two auxotrophic strain and then he cultured this mixture by using a minimal media, and he found growth in that minimal media because in that minimal media a new strain is formed. That strain is actually formed by the genetic exchange between these two auxotrophic strain. So the new strain can synthesize all those nutrients like biotin, phenylalanine, cysteine, threonine, leucine and thiamine. So they are known as prototrophic colonies. That means when mixing these two auxotrophic strains, a genetic transfer between these two bacteria occurred, and that results in the formation of a new strain, and that strain is known as prototrophic strains, and it can synthesize all the essential nutrients required for their growth. So through this experiment, he concluded that genetic transfer, that means genetic transfer is possible between the bacteria whenever they are in direct contact. Actually, Lederberg and Tatum did not directly prove that physical contact of the cells was necessary for gene transfer. Actually, this evidence was provided by another scientist, Bernard Davis, in 1950, who conducted a U-tube experiment. For the experiment, first of all, he created a U-tube. This U-tube is consisting of two pieces of curved glass tubes. They are fused together at the base to form a U-shaped tube. Okay, so this U-shaped tube at their middle, which contain a filtered glass or glass filter, and this glass filter divide this U-tube into two halves. The filter allow the passage of media but not the bacteria. The U-tube was filled with a nutrient medium and each side inoculated with different auxotrophic strains of E. coli. During incubation, the medium was pumped back and forth through the filter to ensure medium exchange between the two halves occurs. After 4 hours of incubation, the bacteria were plated on minimal medium. Davis discovered that when the two auxotrophic strains were separated from each other by the fine filter, gene transfer could not take place. Therefore, direct contact was required for the genetic transfer between the bacteria that Lederberg and Tatum had observed. And whenever this glass filter is removed from the U-tube, now the two auxotrophic strain in the either side of the U-tube can mix each other and they will be in direct contact and they transfer their genetic material, which results in the formation of a new prototrophic colony. And later, if we draw sample from the U-tube and if we cultured it in a minimal medium, we will get colonies because their prototrophic colonies were developed.